Welcome to the Molecular Biology lecture for the 3rd of February 2017. This is really beginning to feel more like an online class, or at least a hybrid class as far as lectures are concerned. Today, just wanted to start out talking a little bit about the exam. Yes, I was planning on giving you back your Scantrons today. Assuming we have school on Monday, which looks like we will, I will have them then. So without any further ado, this is a reminder of how I set up grading. I only assign letter grades at the very end of the class. So I add up all 3 of your exam scores. Then I take the 10% for the clicker scores. All of those get normalized to the high score. And then I just go through and say 90% of that score is an A, higher than 85% is an A minus, etc. All the way through. This is very different than the grading they give you on D2L. Just ignore what you see on D2L. I know some of you were not as happy as you would like based on the scores that you got on the first exam. I've posted a couple of suggestions from other students who've done well on exams in the past on D2L, so you can take a look at those. There were a couple of questions over from the exam that I wanted to go over in a little bit more detail. And this has to do with things that I evidently think I did a bad job of explaining, but also things that I think could be useful as far as the rest of this course is concerned, and particularly for the cell biology course. And that brings us to this question, which when I bring these up, this means that at least 80% of the people ended up getting them wrong. So that's why I figured it's probably a good idea to go over them again. So this question is, what is the slowest step in a GTP binding protein signaling cycle? GTP binding, GTP release, GTP hydrolysis, GTP binding, or GDP release? The correct answer is GDP release, and that is really based on this slide that we had in the lecture, where what we had on that slide was this G protein cycle down here at the bottom. First, you have a G protein bound to GTP. Usually this is in the active site. At some rate, there will be GTP hydrolysis. And then you have a GDP bound form of your GTPase, which is a different structure. In this case, it's the switch helix moving over a little bit. And this is now inactive. And this is the point that the G protein really can't do anything. It has to have release of this GDP and exchange for GTP. It's this process with the guanosine nucleotide exchange factor, the GEF. This will happen. It will fall off by itself, but it's incredibly slow without that. And you really need these exchange factors to stimulate this process. The reason that I mention this is particularly in cell biology, when you start talking about some of these extra processes, where you're looking at the whole signaling cascade with these various different G proteins. It's that nucleotide exchange, which is really the very slow step. Another question is actually now more important as far as the rest of this class is concerned. What part or ports of an AT base pair is not in the minor groove of B-form DNA? Could have turned it around and say, what's in the major groove? Keto group on T, methyl group on T, amino group on A, or A and B, or B and C. So what are we talking about here? When we say the keto group on T, that is a keto group here, but there's also a keto group down here. Major groove, of course, is up here. Minor groove is down here at the bottom. So what about the methyl group on T? Methyl group on T is here. That's in the major groove. Amino group on A. Here's an amino group on A. That's present in the major groove of DNA. And there's no amino group in the minor groove. GC base pairs, on the other hand, there is an amino group that's sticking out in the minor groove of DNA. And so it's really the structure of these bases that's going to be quite important as far as DNA repair. We'll be talking about a bit later on in today's lecture. Next question, this is where we seem to have the most confusion, which was which of the following proteins are found outside the replication fork? Clamp loader, DNA helicase, DNA polymerase 3, DNA primase, or the sliding clamp. That refers to this figure, which at first you might say, well, hey, all of these guys are right here in the replication fork, which is exactly what this is. But what this doesn't show is the dynamics of replication, whereby your lagging strand polymerase, which is now associated with one of these sliding clamps, once it gets to the end of its Okazaki fragments, the DNA polymerase comes off, but it stays associated with the replication fork. On the other hand, the sliding clamp still sits here. And why is that? The reason that it's sitting there is actually to help the other processing enzymes which are required for getting rid of this primer. In the case of E. coli, that would be DNA polymerase 1 and RNase H. In the case of eukaryotic cells, that would be RNase H and the flap endonuclease, which are involved there. If one of the choices on this last slide were the single-stranded binding protein, that clearly also would be in places that's not quite in the replication fork, because you can see here that it's really in this trombone part of the structure, which is there. Last question that I wanted to talk about this time, which actually was quite a challenging question in any case, is if you have a cell with a DNA polymerase alpha mutation that did not have a proofreading activity, you would expect the mutation rate would be, and the answers were slightly different here, but way higher, slightly higher, no change, slightly lower, or way lower. So this really depends on what DNA polymerase alpha is doing. So DNA polymerase alpha is right here. It's associated with the DNA primase, which makes primers, and then the DNA polymerase alpha will extend those primers. Well, these primers right here, the RNA gets removed. This Pol alpha only goes for a short distance, and then it gets replaced by Pol delta to continue the synthesis of this strand. So of course, it's exactly what happens at the origin of replication. On a leading strand, you have a primer. On a lagging strand, of course, you can have a lot more primers. And the Pol alpha is going to put down a few nucleotides. If it doesn't have proofreading activity, what that means is a few of these nucleotides are going to be incorrect. But since the overall mutation rate due to proofreading is about 1 in 100 nucleotides, a mutation in, say, Pol delta would have 100 times more higher mutation rate. But one in Pol alpha, since it doesn't make that many nucleotides, is going to have a slightly higher mutation rate, because it puts in the wrong nucleotides, but nowhere near as high as one that you would have with lacking proofreading in Pol delta. The higher mutation rates is kind of immaterial. People have more questions about these particular exam questions or other exam questions, please feel free to email me and I will do my best to answer them. Last thing that I wanted to talk about really briefly, just because we didn't get to it last time around, and a lot of people have asked me this in the past, not so much this year, is what about nucleosomes? And so when I mentioned the video that we looked at last time with all these nice replication forks, basically there were no nucleosomes that were associated with it. And nucleosomes, as we know, package DNA, and they package it pretty tightly, but somehow replication forks have to get through this. You got to be able to pull the two DNA strands apart, etc. And as we talked about before, when we talked about histone modification, any kind of modifications that have happened on these histones have to be present there after you have replication as well. And so this is shown, unfortunately, a little bit hard to see here, but all of the dark colors here are the old histones, H2, H2B, H3, and H4. And then the lighter ones, H2, H2B, H3, and H4, are present here. So in some cases, you'll have a brand new nucleosome put together with brand new histones here. But most of the time, or quite a lot of the time, you'll have different other histones that get added. So for instance, here, you've got H3 and H4, which as we talked about, that's how you assemble histones. Those assemble first. And then you've got an old H2A and a new H2B. Down here, we have a case of an old H2A and a new H2B. But basically what this is saying is the nucleosomes here, as soon as the replication fork comes through them, those histones are dissociated but reassociated basically immediately thereafter, right afterwards, sometimes together with the old, all old, sometimes a mixture of old and new, and sometimes all new. And this process happens through these histone chaperones. The name here is really not necessary. One of the things that this means, of course, is you need a lot of histone protein, particularly when you're undergoing replication. Turns out that histones are some of the very few protein coding genes that the cell really needs that there are many copies of, just because they need so many histones whenever they're undergoing replication, and these particular messenger RNAs are particularly not so much transcribed, but translated in the S phase. And we'll talk about this regulation later on. So to finish up with replication, we talked about the origins of replication, the binding of the initiator proteins, DNA A, or the origin recognition complex. But now what happens at the end of replication? We talked about that really briefly for E. coli. E. coli doesn't have anywhere near as much of a problem as we do because it solved this so-called end problem by not having ends, just have a circular genome. And so the polymerases can just go around and around and around. On the other hand, we and all eukaryotes have linear chromosomes. Well, linear chromosomes are great. They're probably easier to package. Splitting them up makes separating them easier, makes recombination easier, but you have a problem. And that is, whenever you replicate a leading and lagging strand, because all of our polymerases, of course, are always going from 5' to 3', your leading strand is fine. You can copy all the way out to the copy at the end of the genome. However, for the lagging strand, you always have to have a primer. And that primer, at the very end, is going to get degraded. Well, the degradation of that primer actually leaves you with a gap at the end because that primer has been degraded. This, in and of itself, is not such a big deal, but the problem is when this piece gets replicated again, it's now a slightly shorter piece. And then it'll be a slightly shorter piece again because this process goes on and on and on. And what happens is, after each round of replication, the ends of your chromosome are going to be getting a little shorter. Of course, this happens at both ends of the chromosome. This is just showing one of them here in this particular process. So this is something which has actually been well known for quite a long time. This so-called degradation, it does look as if the shorter your chromosomes get. It does happen with age. And also in terms of senescence, I don't know how many of you made it. I was going to ask for a call of hands to the biology department seminar on Thursday, which is really interesting, talking about a lot more details in terms of how this end problem is dealt with. Of course, the way that the end problem is dealt with is through telomeres. None of this should be a surprise to any of you. What telomeres are these repeated sequences which get added to the ends of linear chromosomes to avoid this end problem. Now, what makes these repeated sequences at the end of the genome? It's what's something called telomerase. And telomerase is in fact a really interesting enzyme because not only does it have a reverse transcriptase activity, that's what this green protein is here, also known as the TERT protein, but also has a RNA that is part of this enzyme complex. And what that RNA is, it's RNA which provides a copy that the telomerase enzyme can use to undergo reverse transcription. So it's an RNA-dependent DNA polymerase, uses RNA information to make DNA. So in this process, again, you get multiple copies of this sequence, right here, I should say it's the complement of the sequence in the RNA, then at the end of the genome. So what does this do? Ends up making sure that you don't have shortening every round of replication, as long as you have telomerase activity around, but also allows the ends of your genome to be protected. So what does this look like? One way of looking at this is in the electron microscope. Here you can see the end of a linear chromosome, and that end of the linear chromosome actually ends up being a big looped structure. How does this looped structure form? We'll see that in just a second. It turns out that this loop structure can form because you have all these repeated sequences. Well, how do you get all these repeated sequences? Here you have the end of one of your linear chromosomes. The primer has just been removed from this piece. Now you have the telomerase that recognizes this end. That's partly what our seminar speaker was talking about on Thursday. And then using the RNA template, we'll make multiple copies, say the complementary copies to this sequence in the RNA in a 5' to 3' direction, because all polymerases go from 5' to 3'. So it makes a copy, shifts over, makes another copy, shifts over, makes another copy, etc. Now you've got a nice long piece of single-stranded DNA. Now you can have the DNA primase come in and make a primer, which will then get extended by DNA polymerase alpha in this case, because these are eukaryotic systems where you have this. So you end up with multiple copies, but you still end up with a single-stranded end right here of your genome. There's always going to be a little single-stranded bit. And single strands, as we'll see in just a second, particularly the ends of genomes, are really susceptible to getting degraded. Lots and lots of exonucleases that love to chew in at ends. So what happens is this sequence, because it's a repeated sequence, due to the fact it was made by telomerase, this sequence can base pair to this sequence, forming this kind of T-loop structure. So that's diagram down here at the bottom. This is a relatively new review article looking at telomeres. And all that I wanted to show you here is that, yes, you have this T-loop structure. Here's the TERT and the telomerase here. You also have a whole bunch of other proteins, and maybe actually even some extra RNAs that are here. I do not expect you to remember all these names here, but it's a protein RNA complex that's present here at the end of the genome, which is protecting all of these telomeres. There are a couple of nice reviews of telomeres that have come out in the last couple of years. Some of these are freely accessible, not all of them are. This last one down here is a whole special issue talking about telomeres and what's going on with them. One of the things that I forgot to mention about telomeres, and probably the reason for a lot of these proteins that are attached here, is that these ends, as well as being susceptible to exonucleases, also kind of look like a double-stranded DNA break. And as we'll talk about when we talk about our replication systems and DNA repair, that's going to be something which ends up stimulating lots of different repair processes. So, review, this is all the stuff that we had before the midterm. This is just what we had here at the end. And again, any kinds of questions about this, feel free to email me. Now I'd like to switch gears, and again, unfortunately, this is not an in-person class, so I can't show off all of my DNA models because that's what I usually end up doing at this point. This is where I say that, yes, we, at least all cellular organisms, use DNA as our genetic material, but DNA in and of itself is actually a pretty crummy genetic material to use because there's so many different things that can happen to DNA. And what things that are happening are chemical changes that happen to DNA. Well, unfortunately, it's the chemistry of the DNA which is absolutely critical either for the genetic material, the genetic inheritance, so if you change the chemistry, you change the genes, or you change something about the replication process by changing the riboses, changing the backbone, etc. So this is clearly a serious issue. Chemical changes leading to genetic changes. And of course, any of these genetic changes can be inherited. So, talk a little bit more about these different kinds of mutations, also some of which are known as DNA damage. Inherited, this is assuming that you just have a mutation which changes the genetic material. But in many cases, some kinds of changes to the DNA are going to cause problems with replication, cause problems with transcription, i.e. making the messenger RNA, also probably very important for cancer, and potentially also quite important for aging. Now, DNA may be really crummy as a genetic material, but it has a couple of real saving graces. And probably the main one is there are two strands. You always have a backup copy of your genetic information just by having that opposite strand. And of course, if you didn't have mutation, it would be really hard to have evolution take place. So let's look at a few more of these in detail. First, one of the things that makes it really obvious that DNA repair is very important for humans is there are a number of genetic diseases that have been completely linked to problems with DNA repair proteins. And we've actually talked about one of these already. That's the so-called mismatch repair system, which we looked at a little bit earlier on. We talked about replication and knowing which strand that you needed to repair. We talked about colon cancers. These are some of the genes that are important in that process. Probably some of the best known and best understood genes in terms of DNA repair are what are called the xeroderma pigmentosum gene. There's actually 8 different xeroderma pigmentosum genes. Xeroderma pigmentosum is a genetic disease which causes you to have extreme sensitivity to sunlight, UV sensitivity, and as we'll see, mostly what happens with UV radiation is you end up with DNA damage. That DNA damage gets fixed by nucleotide excision repair. There are a number of other ones down here. One that I did want to mention in particular is Fanconi anemia. There's some really good work that's going on at OHSU and other places now looking at Fanconi anemia, but a number of other genes, even those that are highly mutated in a lot of the inherited cancer diseases, BRCA1 and 2, are important in homologous recombination, which is repairing double-stranded breaks. So that basically the take-home message here is not so much, hey, got to remember all these names for the next exam. It's that a number of inherited human diseases are due to problems with DNA repair, and many of those you see cancer, 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 leukemia, cancer, 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 etc. So, what are the problems that DNA has? Fortunately, most of the time, unless certain people who remain nameless push buttons that we don't want to think about, ionizing radiation is usually not a huge problem, unless, of course, you're trying to treat something which is growing out of control, say cancer. Radiation therapy actually takes advantage of the fact that ionizing radiation breaks the backbone. When I say the backbone, that's your phosphodiester in the DNA, and in that process is being used for cancer treatment. Chemical-based modification, we'll look at this in great detail. This is probably the second most common thing that happens. It's deamination. We'll look at deamination in much more detail. This happens, and it happens all the time, because we happen to like it warm, we happen to like oxygen, and we happen to live in water. And so just because of that, there are lots of changes that happen to our bases, particularly deamination of cytosines, Turns out deamination of cytosines is one of the more commonly inherited forms. Depurination is the most common kind of mutation that happens. In any cell in our body, 5 to 10,000 of our purines are lost just due to the fact that we like to live at 37 degrees, or most of our cells at about 37 degrees Celsius. So very, very common process. Again, we like it at a particular temperature, there's lots of oxygen around, and we're in water. On the other hand, most of us like to be outside. UV light is actually really pretty nasty. Most of the time it will form pyrimidine dimers that we'll look at how those get taken care of in just a second. Before that, I want to just mention mutations. There's a little bit of terminology here that I want to make sure that we get right, so-called transitions and transversions. Transitions are the easy, relatively speaking, kinds of changes that take place. A goes to G, G goes to A, and that's just shorthand here. Purine to purine, R to R, T to C, C to T. Why are these the easy ones? That's because you're not changing the structure of your double-stranded DNA so much when you've got a big nucleotide together with a small nucleotide and you exchange out the big ones or little ones. On the other hand, transversions, where you have purines substituting for pyrimidines or vice versa, those cause much bigger changes that take place. So when you have one of these mutations that takes place, it could be just because your proofread isn't working. You end up with the wrong nucleotide put in. That can be repaired. However, if it's not repaired, then you can end up with a mutation which is actually being inherited. So those are just pretty standard. These are normal nucleotides. And so this is how you can really pick up mutations quite easily, just misincorporation. It's that 1 in a billion that happens just due to replication by the polymerase, proofreading of the polymerase, and mismatch repair. On the other hand, as I mentioned before, lots of other things can happen to your nucleotides. The vast majority of these are due to the fact that we like water. Hydrolysis, all these blue arrows here represent hydrolysis. Again, most of our biological reactions are that. The big blue arrows here, you'll notice, are here in the glycosidic bond, which is attaching the ribose to the base. This gets cleaved, you end up with depurination, G's and A's. Another very common occurrence, relatively speaking, in terms of hydrolysis is the deamination of cytosine. Again, water comes in here, you lose this nitric group. But these are by no means the only kinds of chemical changes that can happen. You have oxidative damage. Probably some of the most common oxidative damage is right here, what's called 8-oxo-G. You can also have oxidative damage that happens here. Usually these are through so-called reactive oxygen species, and also methylation as a mutagenic agent. So not in terms of regulation, but in fact in terms of mutations. There are quite a few drugs which actually can cause methylation, and it turns out that cigarette smoke is very good at causing methylation, particularly at some of these nitrogen groups on the purines, but to some extent on the pyrimidines as well. So this is an overview of some of those. I like the previous slide because it actually has the individual ones. But here, deamination, oxidation, alkylation, and alkylation is often going to be methylation that takes place. But let's concentrate on some of the common ones now. First one is here, deamination of cytosine. What happens when you lose this amine group on cytosine? You end up with uracil, which is a normal base in RNA, but not a normal base in DNA. The most common is this depurination. You have, again, hydrolysis. You just lose the whole base from your backbone. And then this process down here at the bottom, which is a really interesting one, deamination of a nucleotide called 5-methylcytosine. Now, 5-methylcytosine, we'll see later on when we look at regulation, particularly of transcription, but also gene silencing, is a pretty common kind of modification that happens to DNA. If you have deamination of 5-methylcytosine, you end up with thymidine. Well, this is a problem because the repair machinery doesn't know that there's something that's wrong when there's a thymidine here. Whereas in this case, it actually does. The other kind of quite common changes that happen, again, it's much better to have my 3D model for this, is so-called pyrimidine dimer formation. Here's a thymine dimer. It's when two of these pyrimidines stack on top of each other. UV irradiation comes in and forms a cyclobutane ring. What that does is it pulls the two bases directly on top of each other and thereby causes a big problem. Also, as you can see here, they're not going to be able to base pair appropriately with the DNA. So again, thymidine dimer formation. Actually, this can also work with cytosines. It's just a pyrimidine dimer formation. You have deamination that takes place here from your deamination here in your cytosine. Normal cytosine loses that amino group. You end up with uracil. Guanine or adenine is lost through hydrolysis. You end up with your normal DNA backbone, but no purine on your sugar. So how do these get dealt with? Couple of different ways. Probably the most efficient way to deal with particularly pyrimidine dimers is a protein called Photolyase. Unfortunately, humans don't have a nice active Photolyase, but a number of bacteria do. And this is a specific enzyme that will recognize the structure that happens when you have a pyrimidine dimer formation. And in the presence of lower wavelength light, actually reverses this cyclobutane dimer, which is formed here. And this is a great process, not surprisingly. You see it quite commonly in organisms which are photosynthetic organisms because they're, of course, interacting with UV light very often. We, however, don't have a photolyase in human cells, so therefore we have to use different mechanisms. We talked about mismatch repair before. This is actually exactly the same process that we looked at before. Again, for E. coli, how do you know which strand is the one you want to repair? But here, down at the bottom, what I wanted to mention is the process on how this happens. First, you have binding to a different kind of structure, which might sound familiar from the exam, by these mutation detection proteins. And here the mut-S protein, then the mut-L protein. The mut-L protein makes a cut in this strand, which should be on the opposite side now of where this change is. So there's an extra cut which happens here. Now, before you have the strand removal step, you have a piece of DNA that's cut on the outside of this piece that you need to repair and somewhere else. And if you just have the mismatch, this protein, the mut-L protein, will actually cut that. But then you need a helicase to remove this extra single strand, and then all the DNA polymerase activity together with the DNA ligase to put this back together. So that's mismatch repair. Mismatch repair, again, is just due to the base pairs that don't match with each other and the wrong structure. A very similar process happens in human cells and many other cells when they have big kinds of DNA damage that take place that really change the structure of DNA a lot. And so that's what's called the nucleotide excision repair pathway. So the nucleotide excision repair will notice not that you have a pyrimidine dimer here, but it will see that there's some change in the structure of DNA. You remember the pyrimidine dimers? That was stacking these two on top of each other, changing the structure of DNA. Big change in the structure of DNA. The repair enzymes see this, and they make one cut on one side of this, another cut on the other side of it, DNA helicase, which will remove that piece of DNA. Now you have, I'm going to label it on here, but a 3' OH with a primer provided by the other DNA. DNA polymerase can extend along here and hook up to the other end. This is a very general process. Again, it doesn't recognize a particular change here. It just recognizes a structure. It's also pretty inefficient because you have to replicate all of this DNA here. On the other hand, if you have a very specific kind of mutation, and particularly mutations that happen very often, like a deaminated cytosine or a depurination which has taken place. In this case, we now have a much more specific way of dealing with this DNA damage. This is called base excision repair because it's what's happening at a single base as opposed to nucleotides with their multiple ones. So here, a deaminated cytosine, that gives you a uracil. We don't want uracil in DNA, and if we know if we see, to totally over-anthropomorphize again, a uracil present in DNA, that means, probably, it's a deaminated cytosine. Deaminated cytosine, we need to get rid of. So there's a very specific enzyme called uracil DNA glycosylase that will recognize U's in DNA and cut the U off through hydrolysis at the glycosidic bond. Once it's hydrolyzed, this is an apurinic or apyridimic site. It's just missing a base. So now you have an AP endonuclease. Now, just as a reminder, endonucleases cut in the middle of DNA. Exonucleases cut in from the end. So the endonuclease cuts in here. And then a phosphodiesterase, again, it's an enzyme. What does it do? It gets rid of the phosphodiester. So now you have a single base which has been removed. Now, this is in the same orientation here. 3' OH, DNA polymerase, puts in one nucleotide, the ligase will repair it. So this is the very specific process. This is the very general process over here. Let's look at that in a little bit more detail. Deamination, how does this happen? It seems to me to be pretty amazing that there is an enzyme that's just running around looking for the wrong kind of bases which are formed, say through deamination of cytosine. Turns out you can have deamination of other bases as well that give you these other unnatural bases. Any of these that have a amino group that can be deaminated. So there are specific enzymes that cruise through the genome looking at all of the individual bases. So how do they do that? Turns out that bases are pretty floppy. And the enzymes are just scanning along the DNA all the time looking for the wrong nucleotides. Wrong nucleotides just being one of these unnatural DNA bases, which is probably formed through this deamination process, and the best understood of these are the uracils. Again, we mentioned before, 5-methylcytosine, you get rid of it, you have thymine. Well, you can't have one of these scanning enzymes that's looking for the wrong base because thymine is the right kind of base. So this is where you end up getting some mistakes. Again, to risk beating the dead horse here, base excision repair, first thing that happens, you have an enzyme that recognizes the very specific change which has happened here, and now we'll cut off what's seen in red here, the particular base. So here, your glycosylase will cut this off. Once you have the glycosylase, which has removed this, now you have an OH at the 1' position on your ribose. Then you have an endonuclease, which cuts here, and then the phosphodiesterase which removes here. It's now an endonuclease, which turns to an exonuclease. Now you have one base missing. That then gets repaired. Nucleotide excision repair is very well understood, actually, both in human and bacterial cells. But the bacterial cells are probably the best understood of all of these. And not surprisingly, the names for a lot of these enzymes, quite similar actually to what you saw in Xeroderma pigmentosum, are named after mutant phenotypes. So you remember mut-S was an enzyme when it was mutated, you had higher mutation rates. Here, UVR, so these are UV-sensitive bacteria. They're mutated in UVR A, B, and C. UVR A and B bind to a change in your DNA. Again, it's recognizing a structure, not that there's a particular kind of change there. Very often, probably the most common of these changes are these pyrimidine dimers, which are formed due to UV irradiation. So these proteins bind, they cause a change in the structure of the DNA, which allows you to have an endonuclease that now cuts on either side of where this change is, and then a DNA helicase removes that piece. Now you've got your standard DNA polymerase, your DNA ligase, which is going to repair this whole process. Again, nucleotide excision repair. One of the things that people found quite early on in looking at these DNA repair processes was that genes that were being actively transcribed also got repaired much more rapidly than genes that weren't being transcribed. Now, transcription, again, is making a messenger RNA copy. This makes total sense because if you're transcribing a gene and there's a mutation in it, particularly a mutation which causes a problem with the RNA polymerase moving through that particular piece of DNA, then you want to repair that particular problem sooner than some of the other problems. And the way that this works is not terribly surprising, is that you have your UVR A and B proteins, which are actually associated with the RNA polymerase as it's transcribing, such that when it reaches a point where it stops, because it can't get any further, because of this change, which is present in the DNA here, then these repair proteins are at the right place at exactly the right time. Turns out this is exactly what happens with eukaryotic RNA polymerase, with the eukaryotic enzymes that are involved in this repair process. So now I wanted to talk a little bit about double-stranded break repair. So we mentioned already ionizing radiation is one way that this can happen. X-rays, nuclear reactions, etc. But also whenever you're trying to undergo radiation therapy to try and block the activity of cancer cells. Because enough radiation, the DNA gets so broken down that it can't be repaired, and those cancer cells particularly die. Turns out there are also some chemotherapy drugs which lead to formation of these double-stranded breaks. And so when I say double-stranded breaks, that really means both strands are broken at the same time. There are two basic ways that this can be repaired. There's the quick and dirty method, known as non-homologous end joining, or the slow but precise mechanism known as homologous recombination. One of the things I just needed to remind myself here, you remember we talked about the telomeres. Telomeres are just the ends of your genomes. Well, if telomeres were detected as being double-stranded breaks, basically what they are, they're just two ends, then you could have all kinds of problems of chromosomes getting rearranged, etc. So it turns out that telomeres are not substrates for homologous recombination, and partly all those extra proteins that we looked at the telomeres before are responsible for that. Double-stranded breaks in and of themselves, if you have enough of them, you can use them to treat cancer. But in other cases, they can actually lead to cancer. If you remember some of those karyotypes that we looked at earlier on in the course, when we were looking at these genomes, and particularly bizarre are some of the karyotypes that come to these cancer cells, and very often there are many double-stranded breaks which are present there. Double-stranded breaks are actually particularly dangerous in terms of changing your genetic material, and the main reason there is you've lost your redundancy. You remember the saving grace of double-stranded DNA is that you had both strands which were there, and you could use both strands in order to repair the DNA on one strand using the other strand. Base excision repair, mismatch repair, nucleotide excision repair, all are dependent on having that other strand. Double-stranded break, that's a problem. Fortunately, at least we and many other organisms are diploid, i.e. they have two copies of the double-stranded of DNA, and that's where they can get the information from. And so we'll take a look at that. Turns out that's really important for homologous recombination and getting homologous recombination to work. So this is the overview of that process. Non-homologous end joining, you have a double-stranded break which takes place. What happens in non-homologous end joining is the two ends here, they're almost never do you have a double-stranded break which is exactly opposite, one base opposite the other. So these end up getting chewed up a little bit, and then after they've been chewed up and made nice, basically, those two ends get put back together. So this end is not homologous to that end. Non-homologous end joining is putting them together. One of the things that happens here is you have little deletions that take place. Anytime that you're chewing in from the ends here, you end up with deletion of DNA sequence. Turns out that it was this mechanism right here, non-homologous end joining, which was exactly what the original people developing CRISPR gene editing was using. Anytime you have this double-stranded break, leads to non-homologous end joining, causes a problem in terms of losing little bits of DNA, and if this double-stranded break happens in the right place, now you can have specific loss of genetic information. We'll talk more about that when we talk about CRISPR a little bit later on. On the other hand, homologous recombination, now you have two homologous chromosomes. And remember, homologous coming from a common ancestor, in this case, the ancestors of, in a sexually reproducing organism, mom and dad. These are similar to each other, but they're not identical to each other. Here we have a double-stranded break. Hopefully it doesn't happen in both of your sister chromatids. So here, one of your haploid chromosomes, you have a double-stranded break. You have loss of the nucleotides at the end, happens both cases. But now, instead of just popping these two ends back together, now you use this diploid as your backup in order to repair the DNA here on the other side of your piece. So we'll talk about homologous end joining first. Non-homologous end joining. This is the quick and dirty mechanism. Again, double-stranded break that happens in your DNA. This double-stranded break is recognized by a protein called Ku. Ku is a heterodimer. That means it's got two proteins that are not identical to each other. You can tell, green, dark green, and light green. You can actually see the structure down here. These proteins bind to cut DNA ends. Turns out they also associate with telomeres, as we heard about yesterday at our seminar. Once you have Q protein binds, then it recruits a kinase protein. That kinase protein is DNA-dependent protein kinase that also will phosphorylate proteins and DNA. Then you have ligation that happens. These come together and you have a small deletion in your DNA. So particular ligase that does this is DNA ligase 5, or sorry, DNA ligase 4. Just want to remind you there are multiple different ligases that are present in cells that do these kinds of things. So this process, losing a little piece of DNA, again, is really useful for CRISPR gene editing technology. Turns out that an extremely similar process happens in formation of antibodies because antibodies have to have huge amounts of diversity. How do you get some of that diversity? It turns out using a very similar kind of process of chewing up the DNA in order to get variations that happen in your antibody genes, which are happening right here. I think that instead of trying to move forward and talk more about homologous recombination, just finish up here really quickly with how homologous recombination is also thought to work in systems where you only have a haploid genome. And basically, the way that that is thought to work is that you have DNA replication, which is actually ongoing almost all the time, even in something like E. coli. You have a replication fork. This replication fork ends up going along the DNA, and then it runs into what's called either DNA nick or a single-stranded break, where one of the strands is actually broken. What happens now is you have what's called a broken replication fork, because now your leading strand gets to the end here. This now is a double-stranded break, but it can be repaired due to the presence of this homologous chromosome that's just been replicated. How that works, turns out, is exactly how homologous recombination works. And homologous recombination, we'll talk about the processes on Monday.